The overall goal of this procedure is to observe the responses of gustatory neurons in insects and to measure gene expression in gustatory tissues. This is accomplished by first recording the responses of specific gustatory hairs to several tastants at different concentrations. The second step is to carefully dissect the gustatory tissues, then separate the samples by sex and appendage. Next, isolate the total RNA from each tissue sample and create cDNA preparations for next-gen sequencing and qPCR. The final step is to analyze the gene expression data and form hypotheses, such as how the expressed chemosensory genes mediate physiological responses to tastants in insects. Ultimately, electrophysiological recordings are used to show the receptive range of the insect gustatory system, and RNA sequencing of gustatory tissue is used to highlight the most important genetic components of the gustatory reception.
Generally, individuals new to these methods will struggle because of the small size of insect chemosensory appendages. Whether dissecting or recording from small insect organs, practice and patience are the keys to mastering these techniques.
To begin this procedure, pull a thin-tipped recording electrode in a pipette puller with a glass capillary. Under the dissecting microscope, carefully break the electrode tip off so that its opening is wide enough to envelop the target sensillum, but small enough to avoid contacting the neighboring structures.